Species are not discovered in the field, they are in fact discovered in a museum. This is Archbold's Natural History Museum. Today, let's use Archbold's Natural History collection to understand variation at different taxonomic levels. Here are two kinds of owl species. One is the barn owl. These are both Florida native owl species. The underwings of this owl are especially white, so they look really white in flight. This, on the other hand, is a screech owl. Eastern screech owls live all over the east coast of North America and can be this beautiful ruddy color. They live in forests and eat little bugs and frogs and maybe even little birds. Natural history collections where you can have specimens from different parts of the country in the same space lets you compare variation within a species. Here's a common American woodpecker called the northern flicker. But if you look at their tails, they look very different. Here's a red-shafted flicker from the west coast, and they have this beautiful red coloration, while a golden or yellow-shafted flicker from the east coast has golden or yellow tail feathers right here. Another type of variation can be variation in individuals that live in the same populations. In some populations of eastern screech owls, again, these owls that are our neighbors here in Florida, you might have individuals that look very different in color. So here's a gray morph of a screech owl, and here's a red morph of a screech owl. And you might get these two morphs in the same population in the same forest. Some of the variation within a species can actually be really subtle and really difficult for us to see. There might be differences in the beak shapes or the amount of blue in these Florida scrub jay specimens that are really difficult for us to see, but if we measure them right, you might be able to discern differences between these specimens. Another type of variation that is not in physical museums are sound libraries. So the Macaulay Library at the Cornell Lab of Ornithology actually stores sounds, and then you can also compare songs and calls of different birds from across their species range to understand how the calls and songs vary within a species as well. Museums are like libraries for biology. Just like a good library has books from all over the world, but also has all the different editions of every book, a good museum has species and specimens from all over the world, but they also have representation of different populations and different amounts of variation within a species.
This is blue curls, arguably the most beautiful flower that we have here in the Florida scrub. So in rosemary scrub habitat, we have a lot of species that are found here on the Lake Wales Ridge and nowhere else. But the interesting thing about blue curls is that we actually find it all over in Florida in sandy habitats. The question is there only one species of blue curls like we've always thought? Or are there actually lots of species of blue curls, including a new one here on the Lake Wales Ridge? These are things that we might suspect in the field, but to confirm them, we have to go back to the lab. In our herbarium, we have 5,000 vouchered specimens representing the flora of our region. We have 6 blue curl specimens. From these specimens, we can measure traits that might show us the important variation to distinguishing new species. But we only have 6 specimens, so what we have to do is combine forces with other herbarium at universities and museums to try to parse out which species might be which. That's exactly what one team of researchers did. They looked at variation in traits across specimens collected throughout Florida. The research team answered the question and discovered that yes, we have our own distinct species of blue curls here on the Lake Wales Ridge. This really shows that species are suspected in the field, but they are really identified in the lab.